Hello, everyone, and thank you for attending RRS's webinar on flash radiotherapy, what we know and where we need to go, with Drs. Charles Lamoli and Marie-Catherine Vozenin. Questions will be addressed at the end of the presentation with a Q&A, so please submit your questions using the chat box on the right side of your screen. Please note this session is being recorded. All registrants can expect to receive the recording within 2 to 3 business days via email. Moderating today is Dr. Pierre Montegruel. Dr. Montegruel is one of the pioneers studying flash radiation. He began his work at the University of Lausanne, Switzerland, in Dr. Vozenin's lab. There, he studied flash effects on the brain and glioblastoma in mice. He is now expanding his research at the University of California, Irvine, with Dr. Lamoli. Welcome, Dr. Montegruel. I'll hand it over to you.
Thank you, Lindsay. I hope everything is doing well. Good morning, friends and colleagues. Good evening if you are connected from the old continent. I am very happy today to present this new webinar from the Radiation Research Society, especially because the two presenters today are scientists that I particularly appreciate. The first one is Dr. Marie-Catherine Vozenin. Marie-Catherine was graduated, had a master in biomedical science and a PhD from 3 prestigious universities in France. She had her first position as a staff scientist and junior group leader also in France, and then was a team leader at the Institut Gustave Roussy in Villejuif, France, before moving to Lausanne in 2013, where she is the head of the radiobiology lab. Since 2019, she's an associate professor in the Department of Radiation Oncology at the University Hospital and University of Lausanne, and she's also an adjunct professor at the University of California, Irvine since 2018. Since the beginning of her career, Marie-Catherine is an expert at deciphering the biological response to radiation therapy of multiple organs and tumor types. She has been an integral to the development of innovative approaches to increase the therapeutical ratio of radiation therapy, and you know her more particularly because she is one of the pioneers in flash irradiation, and she's at the center of a multidisciplinary team in Lausanne, including physicists, physicians, biologists, and chemists that all study flash irradiation. On the other side of the world today in California, we have Dr. Charles Lamoli. Charlie has graduated in chemistry from the Michigan Institute of Technology and has a PhD from the University of California, San Diego. He moved to San Francisco at UCSF for a postdoc, where he had his first academic position for 12 years before moving to Irvine, California in 2006, where he is now the full professor in the Department of Radiation Oncology and a vice chair of academic affairs. Dr. Lamoli's research has focused on radiation effects in the CNS. He has defined many of the mechanisms that contribute to radiation-induced cognitive dysfunction. And he also studied oxidative stress in the brain, disruption of the neural structure and synaptic integrity, and stem cell therapy. He has also studied the effect of cosmic radiation on the CNS with the NASA, and you know him also because since 2016 he works on flash radiation therapy and especially its effect on the brain, from the mechanisms to the cognitive functions. The collaboration between Marie-Catherine and Charlie started in 2016, I think. It's a collaboration that sometimes takes place in the Swiss Alps, sometimes in the US, and they published together 2 papers, 1 scientific papers in PNAS in 2019 and 1 review in 2019, but I can already tell you that many other papers will be published in 2020, so keep posted. And they also were awarded with other labs in the US by a PO1 research program funded by the NIH, program that has been funded after the first application, so it can show you how flash radiation therapy is a hot topic nowadays. So the webinar agenda for today, Marie-Catherine and Charlie will talk about the beam parameters and optimization of those parameters to obtain the flash effect, also the chemistry and redox biology behind the flash effect, how the flash effect is defined, all its application in preclinical and clinical models, the pros and cons of different beam modalities, and of course they will answer all your key questions and everything that still needs to be resolved. So I hope I convinced you that you have the best duo today to talk about flash irradiation, and if I did not, I will leave them convince you. So Charlie and MCAT, the stage is yours. See you later.
Very good. Sorry for the glitch, everyone. So again, we're going to start the presentation off, and I'll present the first part of this, and then I'll turn it over to my esteemed colleague. And we hope you find this informative, and we'll do our best to go through this very slowly and carefully. And as you may or may not know, there's a lot of excitement about this flash radiotherapy, which is really an ultra high dose radiation modality compared to what we normally use in the clinic and in most of our labs. And many groups have now published on this since really kind of a renaissance paper that Dr. Vozenin published with Dr. Favadone in 2014 in Science Translational Medicine. And you can see here on the left, the blue references are really from Dr. Vozenin's group, and the other groups that are publishing are shown in the black here. And what these have basically described is a lot of normal tissue sparing in a variety of preclinical models. So as we go through this presentation, we'd like to touch on some of the highlights of this work. Next slide. And so one of the things, of course, that was first and foremost the question to all of those thinking about this normal tissue sparing, well, what good is flash if we're going to spare the tumor? And luckily for us and for hopefully the field of radiation oncology, we found that flash radiotherapy was actually iso-efficient compared to conventional dose radiation modalities at controlling tumor growth. And again, the papers on the left and the right are publications from Dr. Vozenin's group and some other people showing that, in fact, using either protons or electrons, that we can control the tumor equally well, and we'll present data further on this. And so what's really at the heart of all this now is why exactly does flash or ultra high dose radiotherapy spare the normal tissue and yet equally efficiently kill the tumor? So next slide. So one of the things that we need to point out is that as we all try to wrap our minds around this flash technology, we need to keep in mind some of the important technological features of this technology. And one of the things I'd like to point out, and what you see here is the ERT6 Ariatron electron LINAC that is really in many ways shaped our field today. More publications have come out of the use of this machine for flash than anywhere else. And one of the distinguishing features of this is that you have to realize this is not a continuous beam, it's a pulse beam. So right off the bat, that defines a couple of important critical parameters. One, if you look at the diagram here on the right, you can change the frequency of the pulses that are delivered over a total duration of exposure. And within this frequency or total duration of exposures, you have these pulses. They can have different fluences of electrons or X-rays, and so this defines what we call the intrapulse dose rate. So these two parameters are important to define, and of course there's been a significant effort from Dr. Vozenin's group, really led by Dr. Ballet here, and some of the other physicists shown here have played an integral role in really defining the dosimetry, because if we're going to convince people that we can do these flash studies, we need to convince people that we can measure the doses accurately at the really high dose rates. And they've done a very nice job measuring the dosimetry through multiple mechanisms. And so now let's go to the next slide. What this slide really points out now is some of the things we need to think about moving forward. So what we have here on the y-axis is the total radiation time required to deliver a 10-grade total dose. And on the x-axis, we have the dose rate in the pulse. And so in the center of the plot here, we can see a series of single pulse studies, largely from the groups in Europe. And then we can see some multi-dose studies where the total dose was delivered from 3 to 11 pulses. And we can see that the average dose rate per pulse there is about 1 million, about 10 to the 6th gray per second. We can see on the lower right a unit that doesn't exist anymore. The Febitron was able to give a single pulse very quickly. And then we have one experiment over here where we use synchrotron X-rays where the total duration in exposures was relatively high, but the dose rate was more on the order of 10 to the 4th per gray per second. So all of these experiments shown in this plot now have been able to demonstrate the flash effect when these total doses were delivered with this number of pulses at these intrapulse dose rates. So you're thinking, great, the flash effect is something that's robust, we could easily generate it. Not so fast. Let's see the next slide. So what this shows here there are some reports in the literature where under similar conditions where you see the flash effect, you can see on the right here, we don't see the flash effect. So there's other things we need to consider. If we look at the multi-pulse studies here, we can see in working with the ERT6, if we lower the intrapulse dose rate, these X's represent no flash effect. We can see at least under the conditions with the 5 to 6 MeV electrons, we start to lose the flash effect when the intrapulse rate drops. We can also see over here, there's a report now with protons from UPenn and another report from the proton in Oncoray in Dresden, where under very similar conditions, we either do or we don't get a flash effect under very similar intrapulse dose rates. One could argue maybe you're at the threshold here, but as you can see, you need to be careful when you start to define your beam parameters, and this is the main point of this slide. If you're going to report on the flash effect, I think it's very important for the field to see precisely what your beam parameters are so that we can place you on this chart. If obviously you're using electrons and you're using intrapulse dose rates down here, the data seems to indicate you're not going to be in the range that you need in terms of intrapulse dose rates to get the flash effect. Clearly, there's more work we need to do in characterizing these beam parameters, but these are important considerations for all of you thinking about getting into the area of flash radiobiology. Okay, next slide. So another question that is often asked to myself and Dr. Vozenin, among others, is what is the mechanism? We get to deliver this radiation in milliseconds, microseconds, and why is it so different than when we deliver this radiation instead of tenths of seconds over the course of seconds to minutes? So the short answer to this is we don't know. We have some ideas of what the mechanism are, and I'd like to go over some of those ideas with you. So next slide. One of the more interesting experiments that we published in Pierre's paper in PNAS was we have a neurocognitive test that we call the novel object recognition task, and this is simply a test that measures the ability of an animal to discriminate novelty. And we quantify that by this term on the y-axis that we call a discrimination index. So the higher the value on the discrimination index, the better the animal performs on that particular task, which in this case is the novel object recognition task. So as we can see here, our controls, right, do very well. They're right around 60 for this discrimination index. So what we did in this experiment is we gave animals carbogen. We let them breathe it before and during the irradiation. And basically what the carbogen does is doubles the PO2 of the oxygen in the brain. So let's see what happened. So we took these animals that were exposed, in this case here, either to conventional irradiation in the middle, where animals were given normal breathing or carbogen breathing. And we can see, as we found before, that the 10-grade dose of irradiation significantly compromised their ability to distinguish novelty on this task. When we gave animals the 10-grade flash under normoxia breathing conditions, we get our neurocognitive sparing that you've come to recognize as one of the signature effects of the flash effect in the brain. However, when the animals that were given carbogen were given the same test, increase in the oxygen dropped the capability of flash to preserve neurocognition. So one could assume, at least from this, that oxygen at least plays some role in impacting the radioprotective effects of flash. And so when we increase the oxygen concentration in the brain, we lose the flash effect. All right, next slide. So this led us to propose at least one model that has stimulated, at least I can say, some level of controversy in the field, but that's good. Everyone's thinking about it. So one idea, at least one explanation that can at least support that observation, is that there's a transient radiation-induced hypoxia that protects only normoxic tissues. And so if you hit the button, we can animate this slide. There we go, and keep hitting it. So what we do in the conventional irradiation, we produce lots of reactive oxygen species that lead to brain toxicity, all right? And then the brain toxicity is a cause of problem. And then now in the flash radiation situation, we produce much less ROS as shown here, and we get a transient protective hypoxia, and we get less toxicity. So this idea of adding extra oxygen to the organ during flash and eliminating the flash effect is something that requires some additional experimental validation, but it's at least supportive in the sense that oxygen seems to play a role in mediating some of the downstream effects of flash radiotherapy. All right, next slide. So that, of course, doesn't explain everything. In the group in Iowa, Doug Spitz and Gary Buettner got together with me and Mary Catherine, and we've had these discussions with Jolie and Henry. And here's a model that Doug has really championed in the field now. And this at least provides at least a partial explanation. At least this provides something that we can formulate some experiments to test these hypotheses. And what this graph shows you is that on the left, we have a flash pulse that over a very short amount of time produces orders of magnitude more free radicals and organic hydroperoxides than a conventional pulse that's delivered over the course of seconds to minutes, all right? So on the right, we have a conventional pulse. On the left, we have a flash pulse. The first thing you're going to notice, you're never going to uncover any of these differences in the normal and tumor tissue that you will under a conventional dose rate. You have to saturate this system with the flash pulse. At least this is our idea. And what you see here, look, given a flash pulse, there's going to be the same amount of radicals produced in the normal tissue and the tumor tissue. So what do you get? Why is the normal tissue spared? So the idea here is the organic hydroperoxides in the normal tissue are much more efficiently removed. And in the tumor tissue, the half-life of these is much more persistent. So the free radicals do not get removed as quickly in the tumor tissue. We believe this is in part due to a lot of labile iron in the tumor tissue that promotes a lot of Fenton chemistry and promotes a lot of free radical chain reactions that continue and persist the half-life of these radicals. So it's the difference in the slope, in the backward part of these lines, that then we believe defines the therapeutic index. Again, you're not going to see this effect unless you saturate the system. And we think it's really this instantaneous burst of free radicals. The tumors just can't deal with this the same way the normal tissue does. And so we get, in the end, much more effective tumor killing after a flash, whereas the normal tissue can compensate and cope with these organic hydroperoxides. Now, whether this is a complete or partial explanation of the mechanism obviously remains to be seen, but it lets people formulate some experiments again to test to the extent that this is accurate or at least partially accurate. So, moving on from here, next slide. So, we obviously need to run many more experiments. You know, our groups and many groups around the world, I'm sure, are actively thinking about this. We have a lot of work to do. So, I mean, there's obviously changes to physical chemical event. We seem to implicate oxygen is important in this event. There's a lot of work we need to do to really pin down whether, in fact, the DNA damages response is different between these irradiation modalities. We need to look at whether the molecular signaling pathways are different, which I think we can assume they will be different. There's going to be the expression of different cytokines, different levels of acute and persistent oxidative stress that we induce. There's obviously going to be a big difference in how the immune systems respond. We've already pointed to differences between neuroinflammation, and all of this combines to give us an anti-tumor effect in the functional preservation of normal tissue following ultra-high dose or conventional radiation regimes. So now, it may seem like we really know nothing about the mechanism, but actually, if we go to the next slide, we're starting to piece together some of the things that we do know about the biological mechanisms in normal tissue. So next slide. So what we know is that in a recent paper from Christoph Peterson's group and our group, we do, after flash, get lower productions of hydrogen peroxide. And we also have implicated the importance of oxygen in this effect. In a recent paper in Clinical Cancer Research from Foyad et al., Well, they found some interesting data suggesting that after flash, there's lower levels of persistent DNA damage in senescent cells in the lung. As I highlighted in the prior slide, we speculate, at least for now, that the metabolism and redox free radical chemistry is going to be different after a flash versus a conventional radiation dose. Our group, along with Dr. Favadon's group in the brain and the lung, have really pointed to differential immune responses and inflammatory signaling in those tissues. And we also have pointed to there's some different impacts of signaling and stem cell protection. And so these are little bits of information now that have been published that are starting to give us a hint about going from the initial radiation chemistry down to the preservation of normal tissue and the isoefficiency of tumor kill. We're starting to piece together some of the events that take us from the start to the end. So now I'd like to share with you some unpublished data from our groups. Next slide. And this hopefully can stimulate some further interest in the field about what flash may or may not be doing. There have not been to date a lot of reports on how flash or conventional might impact the vasculature. And this is some data that's currently under review from our group showing that in the hippocampus, what we've done here is we've injected mice following a radiation with flash or conventional. And so for most of the plots that are coming up, the first bar you're going to see is going to be a control. In the middle, we're always going to put the conventional radiation modality, and on the right, we're going to put the flash radiation modality. And as we can see, after various doses, and we did some higher doses at early times, 25 gray, and we lowered the dose a bit to 10 gray to look at later times. But what we can see is after conventional radiotherapy, if we look at vessel dilation by following the immunofluorescence of a fluorescently labeled lectin that we inject into the vasculature, we can see that the flash radiotherapy really preserves the dilation, and it's much more similar to controls as compared to conventional. And we've also found that this seems to correlate with the reduced expression of eNOS, endothelial nitric oxide synthase, in the flash radiated brain. And so moving forward, next slide. One of the things that we've pointed out in several recent publications, and this is really a lot of the work that Pierre and Marie have done while they've been in Lausanne, is they've really been successful in demonstrating the neuroprotective benefits of flash. And so when Pierre came to the lab and we were thinking about things to do, and one of the questions that I had was, what about the juvenile brain? The juvenile brain for humans, we know that for humans treated with, for example, medulloblastoma, the young brain's a very radiosensitive one, and there's many reasons for that. And so we asked, flash is very good in the adult rodent models. What about, can we preserve neurocognitive function, and can we spare normal tissue damage in a very radiosensitive young animal? And so we set out to do some experiments, and I'm happy to show you a series of studies now. Next slide. Where what we've done now is I'll take you through some of these neurocognitive tests. I won't get into the nitty-gritty details, but again, what you'll see for the bar charts that are coming up, again, control will be in the far left. We're going to be comparing the control radiation, the zero radiation, to the conventional irradiated animal and the flash, which is always going to be conventional in the middle, flash on the right. And what we can see on this task is this is a more complicated version of a novel object recognition task, where in training session A, the animals learn to recognize A1 and A2. In the update session, the animals will learn to recognize that A2 is now switched to A3. And on a test session, they're going to ask, so now A3 has been switched to A4, which is the new novel location. And this is a test that has lots of parallels now with human testing. And it's a nice way to interrogate two memory traces in the same task. And what we can see is if we look at the discrimination index for the update session here, shown in the middle, we can see that the conventional and the flash irradiated animals don't do too well at 2 months, but over the course of time, we can see that the flash irradiated animals come back and the flash preserve this longer term memory trace. And so the reasons for this will become apparent in a few slides, but again, this is a very interesting example of how flash in a very young animal can preserve memory, whereas a conventional, there's a persistent decrement, at least in this one task. If we interrogate these animals with other tasks, next slide, we can see some of the other tests that we published on. And so this is a more traditional version of the novel object recognition task. And again, if we look at the discrimination index, we can see, as you'll see in almost all these slides, the flash irradiated animals are almost identical to the control animals, whereas after conventional irradiation, their ability to discriminate novelty in the novel object recognition task is severely compromised. If we look to the lower right on a social interaction test, and in this test here, irradiated animals are presented with new cage mates, and we videotape these animals to see how much they interact. We can see that after conventional irradiation, the time spent interacting here on the lower left on the part, or the lower left on the right, time spent avoiding. These animals that have been given conventional irradiation seem to be a little anxious, perhaps stressed out, if that's the right word. Whereas again, the flash irradiated and the control irradiated animals on the social interaction test perform statistically indistinguishable. If we then look at an extinction test, now this is a slightly different test that measures the capability of animals to unlearn the association between an innocuous tone and aversive foot shock. This is an active process. It's really important. It's a higher level of cognitive function. Again, we can see that the capability of control in flash animals to reduce their freezing time or actively unlearn this association is comparable, whereas in the control irradiated animals in the middle, they simply can't dissociate this aversive memory. So again, the flash is shown to do very similar things with these neurocognitive tasks than the control irradiated animals. And I can tell you, we have other data. There isn't a single neurocognitive indices that we've tested in these juvenile animals where control and flash didn't behave identically when we compare it to the conventional radiation modalities. So this was some very encouraging data that we got showing that really in the juvenile brain, we could again preserve over the course of 2 to 4 months neurocognitive function after it. And the doses here, by the way, this is an acute single fraction of 8 gray radiation. So let's look at some of the reasons why this may be the case. Next slide. So what we're looking at here, and I'm not sure that the image is coming across too well, but again, after radiation, we're looking at the number of newly born neurons. And so for this, we're looking at the expression of double cortin, which occurs 2 weeks after they're born. And what we can see that along the dentate gyrus, we can see that conventional, as has been published many times before, an acute dose of 8 gray significantly drops the number of double cortin positive immature neurons, whereas in flash, we simply do not get that. So this suggests that the flash irradiation has preserved at least these immature neurons, but to make a more formal assessment and to possibly explain why on the novel location memory and updating task that I showed a few slides back, why the flash animals recover from 2 to 4 months. Next slide. We can see that task is very dependent on hippocampal neurogenesis. And when we look at the number of bromodeoxyuridine cells that co-label with N, this is a more formal assessment of neurogenesis. We can see again that in control irradiated animals and flash irradiated animals, now there's really no difference in the neurogenesis, where conventional drops neurogenesis has been reported for decades. So here in the juvenile animals, a lot of the neurocognitive benefits we see can possibly be attributed to the preservation of neurogenesis in the animals. The benefits don't stop there. Next slide. An interesting piece of data that I'm happy to share with you too is that in these juvenile irradiated mice here, if we look at plasma levels of growth hormone, we don't get the drops after flash irradiation shown here in the levels of growth hormone that we do with conventional, suggesting that again, flash irradiation has multiple benefits and possible long-term benefits for pediatric patients that might need to undergo cranial radiation for tumors. And so I presented some very exciting new data with juvenile mice to you. Next slide. And how does this all fit into the big picture now? And so as we wrestle with the clinical treatment of glioblastoma, where we still have very poor prognoses, we don't have quite the same long-term problems with medulloblastoma. because we're able to get long-term survival. The patients respond. We get about 80% of the pediatric cases that have a mean age of presentation of about 8 years with medulloblastoma. And these patients survive because of their surgical resection, their concurrent chemotherapy, and their cranial spinal radiotherapy. The typical treatments for these patients involve 18 to 24 gray, which is an age-dependent decision that the clinicians make. And they will give that cranial spinal, and then they'll finish up with a boost, usually given in 1.8 to 2 gray fractions, up to and exceeding 50 gray. Obviously, these are big doses, and while the children survive this treatment, if they didn't get the treatment, by the way, obviously they would not survive. Long-term survivors exhibit significant impairments in learning and memory and mood disorders, and we can go on and on about that. There's also increased risk of stroke, cerebrovascular complications, etc. And it's our hope that if we use flash radiotherapy for the treatment of these medulloblastoma patients, we can obviously continue to get the long-term survival, but hopefully in the absence of these long-term normal tissue complications. And so we really think this is one of the main points where flash radiotherapy can be particularly advantageous to the clinic. So now I've talked a lot about some normal tissue benefits, and at this point in time, I'd like to turn the presentation over to Dr. Vozenin, who's going to really touch on some of the finer points of tumor cure. Marie?
Yes, thank you, Charlie. I'm turning my camera on briefly. I know that some of you experienced some sound problem. I hope it's really solved now. So let's go to the next slide, and I'm going to turn my camera off now in order to spare. Charlie showed you working a lot on normal tissue sparing and on the understanding. Well, I don't have any more video. There's something wrong happening. Kathy? Lindsay? Do you...
Marie?
Are you on?
For a moment.
I don't know. I'm not seeing anything anymore. Oh, yeah. Excellent. Good, you're back. So we've been working, as I said, a lot on characterizing the impact of flash radiation on normal tissue, because it was basically at first the observation we had that this very short time of irradiation was able to spare the normal tissue. But of course, we need also to know what has happened at the tumor level. So we have been working further in order to characterize those things, and perhaps in the second part of this talk, some of the questions that have been asked before in the chat is going to be answered at least partially. So we have been working with several types of tumors, and especially, can I have the next slide, please? We have access to human leukemia. So this primary human leukemia were engrafted into immunodeprived animals, and the animals were total body irradiated, either with conventional irradiation or flash, because those animals are very sensitive. We used a total body dose of 4 gray, and you have in the upper side of the slide the survival curves that show in orange that 2 of those primary leukemia are more sensitive to flash irradiation than to conventional irradiation. And conventional irradiation performed pretty poorly in those cells. Whereas in another third primary cell type, we can see that it's a reverse. Conventional irradiation is very efficient in controlling the leukemia, whereas flash irradiation is not. So this gave us a very nice opportunity to look at some susceptibility profile. And what we did was to go back to DNA gene profiling experiments that have been done to characterize those 3 different cell lines, and we did a comparative analysis of the profiling patterns that were found in those cells. And you have, it's a little bit small, but you have in the table the first 19 genes that are found in the 2 susceptible cell line. And one of the first targets that come out is a gene called GAD45 that you probably all know, and that is very relevant to control the cell cycle in p53 dependent pathway. And if we did several types of bioinformatics analysis, you can see that the cell cycle dependent of p53 is really modified in these cells that are sensitive to flash irradiation. And it's true with different type of analysis. Can I go to the next slide, please? We are working further also in order to look at the hypoxic tumor, and I saw that Brian asked a message about this question. What is happening under hypoxia? Can you show the animation, please, Lindsay? In fact, what we did was to do a very simple, no, previous slide, yes, thanks. Very simple experiment. We engrafted U87 in the flank of mouse and did hyperoxygenation either with carbogen or clamped the tumor in order to get to hypoxic status. And you can see on the left side that when you use conventional irradiation, using hyperoxia, we do radiosensitize the tumor. It's a curve, the tumor grows delay in slide blue. But when you do put the tumor under hypoxia, you induce radiation resistance, and it's a purple curve. That's well known. It does reproduce very old data produced in the Gray lab like 40 years ago, so it's very interesting. Now in orange on the right side of the plot, you do have what is happening after flash irradiation. And as we observed in the normal tissue, hyperoxia sensitize the tumors, whether clamping does not. And we do have a superimposition of the flash under normoxic or hypoxic conditions. So this question of hypoxia might be probably much more complex to understand. And we are now doing a lot of profiling analysis in order to identify some potential targets in this setting. But if we are going forward, can you go to the next slide? What we want really is to use flash irradiation in the clinic. And of course, it would be really nice to know all the mechanism and all the biology behind flash RT, but I guess it's going to take us a bit of time to sort all that. It's a base for medicine. We can move forward with a flash into the clinic before we have characterized everything. Given some definition and some important points that needs to be investigated before. First, the technology. Do we have the technology able to go into the clinic? Next slide. Next slide. Yes. So today we have been using a lot this ERT6, which is a LINAC, a very intermediate type of energy, 5 MeV. It's flash validated, it's available. Working with our colleague at the ESRF, Synchrotron, we've seen that photon irradiation was also to be was also able to trigger the flash effect, especially when we used a broad beam irradiation. Recent data showed that the proton, and especially proton delivered in scattered beam and in the plateau phase, are available to produce the flash effect. There is some other developments that could be done with VHE, but nothing is available so far to do at least in vivo experiments. So this is a big limitation. Next slide. And if we are looking at the technologies that I just talked about, you can see that in fact, from the microstructure of the beam, there is a lot of difference in between our electron beam on the far right, the proton beam in the middle, and the synchrotron beam. So there is a huge difference, and this huge difference is also related to an important parameter that needs to be investigated, and that is the scanning process. In our hands, we are not using any scanning. We are able to deliver a big dose of irradiation in a quite large volume, but not very far and deep in the tissue. Whereas with the proton, the structure of the beam require spot scanning in order to be able to irradiate a whole volume. And for the synchrotron, it was pretty much the same. When we did broad beam irradiation, in fact, we scanned the very small beam of the synchrotron over the volume, suggesting that this scanning can perhaps be applied. But it has to be validated. Next slide. So if we go back to our devices and the things that are available today with the low energy electron or intermediate energy electron, we do have flash beam. It's validated with pretty small size of field, like 1 to 2 centimeters, but the limitation is that it's superficial and its flatness is not optimal. For the photon, it's the same. It has been validated broad beam. It has also been validated MRT, but this is another story. The problem with the synchrotron is the synchrotron availability, is the size of the beam, is the spatial fractionation. For the proton, as I said, scattered beam have been validated for flash. PBS still needs to be validated. And here as well, we do have this question of size of the beam and scanning relevance. For the VHE right now, we don't have anything available, but for sure, they are going to be the most efficient in producing ultra high dose rate. Next slide, please. So at the SUV, we do have kind of a pragmatic approach in order to do the clinical translation. We decided that we were not equipped to move into the proton field. We cannot wait for the VHE to be developed, and we don't have any synchrotron available nearby. So we just decided to use some linacs that can be available, and those linacs are derived from the first linac that I showed you, the ERT6, but are now coming with enhanced energy from 8 to 12 MeV. And this pragmatic approach, because those type of energy cannot be used to treat deep-seated tumor, we decided to go for the treatment of superficial tumor, squamous cell carcinoma, basal cell carcinoma, melanoma metastasis, as well as intraoperative setting. And the clinical trial are going to start this year in 2020. Of course, it's a narrow target, it's a narrow group of patients, but it can be used as a proof of principle to demonstrate the superiority of this type of irradiation in the clinic. Next slide, please. And in fact, that's what we did. We treated the first patient 18 months ago now. Well, this was really a feasibility study. The patient was having a very radiation-sensitive tumor, cutaneous lymphoma, and we delivered 15 gray to this lymphoma. We were expecting a very nice response that you can see in the upper left side, sorry. But the most interesting is not really coming from this acute time points. It's coming from the follow-up studies that we are doing. And here I show you a picture of the arm of the patient taken 1 year post-irradiation. And you can see that the flash area with the yellow arrow, the area of the skin of the patient that has been treated with flash radiation therapy appears really normal. Whereas cycle in blue, you do have the previous zones treated with conventional radiation therapy. And you see the very specific type of skin contractions that you have there that is really significant of radiation-induced toxicity produced by conventional dose rate irradiation. Next slide, please. We are moving on further with the clinical trial, and especially we are working with the vets, Carla Rohrer at Zurich Veterinarian Hospital. And we are doing a phase 3 clinical trial in cats with squamous cell carcinoma. We are comparing 2 arms. The first arm is external radiation therapy with the treatments that has been validated by Carla Group. It's an accelerated treatment of 48 gray. And in the second arm, we're using our flash irradiation. And here we deliver 34 gray in a single fraction. So it's a very aggressive type of treatment. The dose is delivered in 3 pulses and 20 milliseconds. It's a single fraction. Next slide. And you can see the first data with the first cats that have been included in the trial. We will have 29 cats included. We're going to be Monday including our 15th cat. So we are expecting the full result to be available in like a year to 18 months from now. But you can see that the outcome of the 2 types of radiation are really good. Mushti at the top of the slide is showing a very nice tumor response with a little bit of swelling in the second week post-irradiation, but overall the tolerance is very good and the anti-tumor outcome really good as well. Sammy, on the other hand, I lost you. I think I lost you. It's okay. It's back. Sorry, I guess it's my internet that is coming back and forth because of the storm here. So in the lower panel, you have Sammy, and Sammy was a lucky one. He was treated with flash irradiation, only 1 session, so means only 1 anesthesia for the cat. And you see that the outcome is really nice as well, and he had really, except the depilation, no side effect. So we're going to be able to compare the anti-tumor outcome to normal tissue toxicity. But if the outcome is similar, it means that flash irradiation for this type of treatment in cats is highly superior than conventional irradiation that require 10 anesthesia and multiple fraction. Next slide, please. We are also trying to go on into intraoperative settings trial using domestic dogs with glioma and meningioma. And the idea here is to do a phase 1 trial with the dose escalation starting at 20-25 gray. But to be honest, I still have not found the funding to do this trial. So the trial is ready. The vet in Bern, who is a vet neurosurgeon, is ready as well. There is quite a lot of dogs with this type of tumors, but we are still searching for money. Next slide. Another very important parameter for the translation into the clinic is, of course, a medical physics question, and it's the development of monitor chamber, which is really mandatory for any kind of clinical application. So this is the work of Claude and Patrick with Raphael, and they are really in the process of being able to propose such a monitor chamber for real-time monitoring of flash irradiation in order to pilot, check, and verify the delivered dose. But this is obviously very, very important. I see that some people do not hear me. Is it okay? It seems that it's not on. Can you hear me again?
We can hear you on the admin side.
Okay. Okay, hopefully it's gonna be back, so I'm gonna move forward. Can you give us the next slide, please? So another important question to be using flash in the clinic is, of course, to be able to investigate and to validate the impact of fractionation and the impact of intervals, because radiation oncology has always been working in a fractionated setting. So next slide. To do this type of experiments, what we did next, yes, was to start with our well-known and well-defined treatment scheme with 10 gray whole brain irradiation in a mouse bearing a tumor. And with this mouse, we've been able to do at the same time the investigation of tumor growth delay, survival, as well as normal tissue toxicity with the cognitive test, as those mice were alive 4 weeks post irradiation. So we started with our very well-characterized dose, 10 gray, and you can see that when we are using flash or conventional irradiation, flash is in orange, conventional is in blue, there is no difference in the tumor growth delay that is induced by irradiation. There is no difference either at the survival level, but we do keep in an animal bearing a tumor the cognitive protections that we saw before in Charlie's part. We increased the dose, single dose, 14 gray, and in that case, we've been able to be a little bit more efficient at the tumor level, but it's still pretty disappointing. The two curves, the blue and the orange, are superimposed for the tumor growth delay. They are superimposed in the survival, and in that case, we are losing the flash protective effect. So meaning that flash cannot do everything. If you do increase the dose, you're gonna hit the toxicity. Now, what we did was to make some fractionation. Next slide. And no, just back. It was just one too quick. Yes. And we did fractionated experiments. We fractionated the dose of 14 gray, 2 times 7 gray. You can see that at the tumor level, it's the same. Flash and conv are doing the same tumor growth delay, same survival, but the 2 times 7 gray does keep the normal tissue sparing. So it means that if we fractionate, mainly hypofractionation here, we can keep our advantage at the normal tissue sparing level. Next slide. We went further and I tried to go as close as possible to clinical scheme. But you can imagine that those animals were bearing a tumor, pretty aggressive in the brain, so it was not possible for us to have an anesthesia more than 4 times. So we decided to use 4 times 3.5 gray. And in that case, again, you see that flash and conv is giving the same tumor growth delay, the same survival. And here, in fact, there is no benefits of flash irradiation at the cognitive level. Conventional irradiation, when used fractionated, is pretty efficient at protecting the cognition in the animals. And in fact, it's the reason why we have been using fractionation all these years to spare the normal tissue. So at conventional fractionated, with conventional fractionated regimen, in fact, there is no real benefits to use flash. The last part of the data I want to show you are really encouraging, and we are working further in that direction. We increased the total dose delivered with hypofractionated scheme and use 3 times 10 gray irradiation. In that case, you can see that the tumor growth delay is much better, and we nearly double the lifespan of those animals. The most interesting is coming again from the cognitive assay, and you see that here, 3 times 10 gray, administered 48 hours apart, that spares the cognition in the normal tissue. Next one. So we have seen that we have some limitation in the technology. We sense that we can use flash irradiation fractionated, especially hypofractionated. We have touched a little bit on the relevance of interval, but this has to be investigated further. And the last big part in radiation oncology is the impact of the volume and the impact and the need of conformality. Is it because we do have flash irradiations that we don't need image-guided radiation therapy? For sure not. So the two have to come together, and so far we don't have the possibility to do that. Next slide. So to move a little bit closer to this conformality, we did a kind of pretty simple type of experiment. We decided to implant in a very specific area the tumor cells in the brain in order to irradiate the animal's hemibrain, flash and conv. And here you can see that the survival data are pretty interesting, either with flash and conventional. So this is probably the way to go. Some more data needs to be produced. We need also to work to investigate the neurocognition in that case. In the mice, it seems that this 25 gray hemibrain is pretty safe, and the mice can cope with it, despite the fact that you can see the discrimination index in the control is pretty low. So we are in the process of performing some more experiments regarding this question of conformality. Next slide. Regarding the volume, it's more difficult, in fact, because investigating the impact of volume in mice is pretty challenging. So what we did, because we had our pig Rosalie, was to try different type of volume of irradiation, and especially a large volume of irradiation. And you see here, we irradiate the shoulder of the pig with a field of 8.8 centimeters. We decided to apply 31 gray in a single fraction, but because our the limitation of our ERT6, in order to deliver this 31 gray in a pretty flat manner, we had to make compromise. So we went up to 20 pulse, which we never do. Charlie showed you that optimally we need to use 10 pulses. And we were pretty slow, only 20 milliseconds of treatment, whereas we like to be under 100 milliseconds. And you can see that in this large field of irradiation, 4 months post-RT, we had a first sign of vascular damage that translated at 5, 6, and 7 months into ulceration. And this ulceration was transient. It healed spontaneously. We did nothing. And at 12 months, we do have a small induration there and a scar at the site of irradiation, meaning that the volume is important and is something that we need to investigate. But if we think about what would have been the necrosis with conventional irradiation, there is nothing to be compared. So the flash irradiation does still spare the normal tissue, but probably we need to optimize the physics parameter in order to be able to treat large volumes. Next slide. Well, in summary, we showed that the tumor growth delay was either efficient when we used conventional and flash irradiation, but that's for sure. TCD studies are required, and we need to go that direction. So this type of studies are pretty difficult to do in Switzerland because of ethics consideration. We have shown also that the flash effect was observed after both single doses and fractionated regimen. We have seen that there is no particular advantage of flash, of using flash when the dose per fraction was low, then conventional was good at it. And therefore, the most relevant regimen in which flash can be used is certainly hypofractionated regimen. Here is the biggest gain in the therapeutic index. Next slide. Okay, with this, I think I'm done, and I have the feeling that we are already pretty late. I really want to thank Charlie's team for this wonderful collaboration. I really want to thank Pierre, who produced almost all the data that I showed you, as well as Benoît Petit, who is really doing all the experimental system in the lab. The physicists at the CHUV are key persons. My friend Claude Bella is really the one that is in charge of all the physics aspects. I want to thank the people from INSEM, especially on the leukemia side. And with that, I think I'm done, and we're going to be happy to answer all the questions that you have.
Yes.
If we can.
Thank you very much for that, Dr. Vozenin, and thanks to everyone involved with this. I think, as you can see, it takes a few people and with many different levels of expertise to kind of move this forward from where we are and hopefully into the clinic soon. Let's have the next slide. We just like to thank the Radiation Research Society for sponsoring this webinar. And of course, as we all know, none of this would be possible without a little bit of money. So we'd like to thank our Swiss and United States funding agencies for the opportunity to try to promote this exciting technology forward. So before we take questions, a few more things. Next slide. Like to thank you again. This is a picture I took of Dr. Vozenin surfing. And we appreciate your time, and we noticed there's quite a few people that were listening. We hope we have provided some useful information. And we're going to move forward with this. So next slide, I'd like to put in a quick pitch and give a quick nod to our council, who's juggling many difficult things in these times, as you know. And I just want to give them a hey-ho right now and let them, I want to pass on an exciting news for next year, and to all of those that have an interest in FLASH, which I think is probably most everyone listening. The Radiation Research Society is currently organizing a winter workshop for early next year. And I'm almost certain that the topics, at least have been agreed to be FLASH radiotherapy, maybe in addition to some other topics. And so the council currently is working out details, largely the date and location. But if you have any requests on potential topics or specific areas of FLASH radiotherapy, or some overlapping scientific disciplines that you'd like to see included in this, please don't be silent and send your suggestions to Audrey, and she will help coordinate your input with the rest of the AMBD team, along with council, as they put together this workshop for us next year. And with that, I think that Pierre will help field some questions for Dr. Vozenin and I. So thanks again for joining us today.
Okay, I hope you can hear me. We've got already many questions. I don't know how much time you want to spend answering questions, but I try to sort them in different topics. So we will start with the physics parameters, maybe. I have a question from Mark Mendonca in Indiana. What are the essential beam and dose rate parameters that should be included in their manuscripts that researchers publish in peer-reviewed journals? I think it's a very interesting question. And if you can be more precise on that, it would be great.
Yeah, I think that in addition to the mean dose rate, as Charlie said, it's really important to quote the structure of the beam, so the frequency, the overall time of irradiation to deliver a given dose, the instantaneous dose rate if the people are using pulsed type of irradiation. So all these parameters, for now, we have not a clear-cut answer of what the most important parameter is. So we need all this information in order to be able to work in a safe way and be sure that we're going to reproduce the data across the world and with all the machines that's going to be available soon. So that's really important. Now we need to move forward as well. We need to extend those parameters, because perhaps we just work with what we could, with what the RT6 was able to do. But there is probably some more possibilities. And regarding what is happening with the proton, perhaps the proton are more efficient. And this has to be very clearly investigated. It's important.
And if I can add to that, you know, as the importance of providing the critical beam parameters are published, I think what we really need to keep in mind is that in the event that people find that they don't get a flash effect, stating the important beam parameters is critical, because we need to know if they were even in the range where this might have been possible. And so, as you can imagine, with any new technology, there'll be some positive and negative reports with this. And if we don't have a clear idea on what the precise experimental parameters were, we can kill this field before it even started. So I think we need to be very careful moving forward that we define things properly. And we'll let the data fall as it may.
And I think that answers another question we got from Amitava Adhikari about a paper published by Zoblinskaya in RadRes in 2014 that showed no observable difference in RBE for pulsed proton beams versus continuous irradiation. So I think it really answers this question. We have two questions about dosimetry. One from Severin Rossum: which detectors have been used for dosimetry? And another one from Dimitris Mihalidis: we do not have dosimetry corrections factor for such high dose per pulse beams like flash. How do you verify that you really deliver what you think you do?
Okay, so basically, all the dosimetry procedure have been published. It's published in Medical Physics, and so everything's available. And I can send a paper if you need. Claude has been developing passive dosimetry because we had, as I said, we had no monitor chamber in this. Connected to this device. So he developed a redundant type of dosimetry traceable to the international standard, and especially in Switzerland through METAS, who is checking all the dosimetry for all the instruments, radiation therapy instruments in the country. So he used this redundant dosimetry with TLD, alanine pellets, gas chromic films, and as well as a Marcus chamber. And for the Marcus chamber, yes, indeed, we need a correction factor. I saw a message around this question of the correction factor. Only for the Marcus chamber, we need a correction factor. And you're going to find that in the literature. It has been published by Christopher Peterson nearly 3 years ago. So yes, there is some need for improvement, and we have in Europe a big consortium that is led by the PTB and Andreas Schuler that aim at defining a robust and suitable dosimetry for ultra-high dose radio radiation. And I think that it's another point that I would like to make. I think that we should not use flash in a kind of general way, as by the way we did a little bit too much, but we should use ultra high dose rates to define the beams. And then the effect is a flash effect. And this effect is the protection of normal tissue and iso-efficacy at the tumor level.
Okay. Then we have many questions on oxygen and free radicals. So one of the first question, and it's very interesting, from Saju Baskaran. How will you account the free radicals? How will you identify them and measure them?
Well, okay.
Is
well,
there a possibility?
You can go for it.
No, I mean, these are studies that are being undertaken. We're going to be doing some free radical chemistry, some EPR measurements, and some work with some fluorescent dyes. But
and spin trapping.
yeah, so I mean, these radiochemical measurements are being done, but the data sets aren't ready for presentation yet. So obviously these are important things to do. So
we then have questions about DNA damage, of course. So radiation therapy in absence of oxygen produces different DNA damages. We know that, like cyclonucleosides. Has anyone looked for these DNA products after flash? And is there evidence that DNA damage processing on effectiveness of flash radiation therapy?
Good question.
Yeah.
Well, we alluded to the one paper in Clinical Cancer Research that's come out. We've got some preliminary data that hasn't pointed to robust differences yet in DNA damage, but obviously the DNA damage products are going to be different. The verdict's still out of whether big differences in DNA damage are a primary driver of the difference in the flash therapeutic index between conventional. But I would have to say that the data in the literature is pretty sparse on that right now, and a lot more needs to be done.
Yeah. Charles Fouillard at Curie just published some data showing the modulation of senescence after flash irradiation, and he has shown very convincing results in normal lung. But there is probably a lot of things that needs to be done at the tumor level, because if we do have less DNA damage, how can we kill the tumors?
Yeah, there's a lot of work we need to do with the double-strand break repair pathway choice, foci, the transfer of phosphate groups from ATM all the way down, and see how the initial changes that involve non-homologous end joining, homologous recombination might differ between the two radiation modalities, as that dictates double strand break repair. But right to the best of my knowledge, those details remain to be elucidated between the two radiation dose rates.
We then have many questions on the paper that you published, Charlie, with dog spits.
Okay.
One from Benaz Bemad. Could you please explain how the Fenton reaction may be different in tumor versus normal tissues?
Sure. If there's more iron available for the Fenton chemistry to occur in a tumor, then that could explain the persistence and constant turnover of hydroxyl radicals that can elicit kill. So the idea is if the iron's not sequestered by transferrin and it's available to react in the +2 state, then when hydrogen peroxide comes in and turns over to make the hydroxyl radical for the Fenton chemistry, the presence of the labile iron promotes chain reactions that occur in the tumor. The lack of that labile iron and the more efficient removal of the hydroperoxides in tumor dictate that the half-life of these radicals will persist less, and therefore the tumor normal tissue spared. At least that's the first pass approximation that we have.
Okay, and we have another remark from Martina Fuss. The hypothesis that you explain in this paper, so the differential redox reaction ensuing the initial radiation chemistry and saturation of the response capacity to hydroxyperoxides, would rather predict a sensitization of the tumor than a radioprotection of the normal tissue. Is there an explanation, and do you think a second effect is counteracting this in both tissues to lead to the observed normal tissue sparing?
That's an awful lot right now.
I agree.
That's a damn good question, and I'm not.
Martina, you were hard on us.
Yeah, I'm not sure that. I mean, the radioprotection of the normal tissue, albeit it's a little harder to explain than the radiosensitization of the tumor. I think that it kind of goes back to my last answer, is that if the normal tissue has less available iron for the Fenton reaction, then there's really less Fenton chemistry going on. These are things that we're actively testing right now, and I hope that these ideas stimulate other people to put this to the test. And in the end, the extent to this is accurate or not remains to be seen. So
yeah, or I could also go into Martina and Emanuele direction. Perhaps it has nothing to do with oxygen.
Yeah.
Indeed.
Yes, imagine that. We in the end, we actually may be wrong about that. So, oh well. I mean, it's
yeah. In fact, that's basically
go.
Yes, that's basically the research. You're making hypotheses. You're trying to test the best you can.
The only way we'll sort this out is for someone to propose something, and if it holds up over the course of time, great. If it doesn't, it'll point us in a different direction. I mean, that's the scientific process. I'm fine with that. So.
Exactly. Biology is not a hard science.
No.
Let's take one more question.
One last question.
One more, and then I think we'll have to send some of them out at a later time.
So maybe we'll go for a question on the oxygen and on the organs. The brain has relative high oxygen consumption compared to other tissues. This is true. Does this imply that flash effects might be more pronounced in the brain than in other normal tissues? And do you think that certain organs could be more responsive to flash irradiation than others? Maybe we have other hypotheses on that, not only oxygen.
Well, I can start, but I think that the response of different organs to flash is, I mean, it's not all going to be the same.
Sure.
And, you know, one of the things to realize about the response in the brain, remember, the brain is half fat, half lipid. And I do think that if there's a lot to do with chain reactions with lipids, then in fact, the brain may be more responsive to those types of things, or at least show a bigger effect to flash than other organs that contain less lipid. Regarding the oxygen, that's a little tougher to call right now. I think we don't know enough to really define which are going to be the most responsive and insensitive tissues. And Dr. Vozenin probably can add to that.
Well, we have been testing a lot of tissues, a lot of different tissues from the gut to the brain to the lung to the skin. And for instance, if you take the skin, it's much more hypoxic. The oxygenation level of the epidermis is pretty low. But still we have this flash effect, and in the peak we have a dose modifying factor around 30% still. So now saying that it's going to be the same in all organs, for sure not. Everything needs to be tested, and I saw a question about what are the lower and higher limits. It's the same. It really depends upon the organ. It depends upon the volume. It depends upon the regimen. So it has to be tested.
But we can point out that, so we have looked at certainly a few organs now, and to the best of my knowledge, the flash has shown beneficial effects in the brain, the lung, the skin, the gut, the bone marrow. And which one am I missing? So certainly not limited to one, but to say that it's equally effective in all is another thing. So.
Yes. I think all the other questions are easy to answer with the chat or with written answers. So thank you for your answers to those challenging questions sometimes, and thank you for your amazing talk.
Okay. Thanks to
okay,
everyone.
Thanks a lot. Thanks everyone for the interest.
Oh, yeah.
Take care. Stay healthy.
Ciao.
Yes, thank you all. We have just a little closing. We definitely want to thank our speakers, Dr. Vozenin, Dr. Lomoli, and Dr. Montegrol for this wonderful and interactive talk. We do still have quite a few questions, so we'll reach out to them to get answers to those for you guys. We will also remember there is the recording coming up, so you'll be able to rewatch and kind of check on some items there. So we also want to tell you guys a little about our future education opportunities. We have several discussion forums open at this time, including the Rad Women in Science, Early Career Investigators, Coronavirus, and soon the RadRes eJournal Club. You can also look forward to webinar recordings and podcast interviews on our YouTube channel, or keep up with the latest news in the field by following us on Facebook, Twitter, or LinkedIn. And last but certainly not least is our wonderful and always updating education center featuring courses, books, articles, and additional learning and teaching materials. Again, thank you all for attending. You will soon receive a recording of this webinar. So have a great day and stay safe.
Ciao.
Bye.
Bye, everyone.
Bye-bye.
Thank you.